My name is Srani. I'm a developmental biologist. I've been working on various aspects of mammalian germline development, which are cells which give rise to sperm and eggs. I was born in Kenya. I was born in a very small town on Lake Victoria. The thing that was of great influence on me was that there were a lot of very exciting fish in the lake. These are called cichlid fish. There are many different varieties. And that was kind of sparked an interest in trying to understand how living things develop. Key always is to find a question. You have to find a question and then also to ask, why is this question important? This I found kind of amazing, that what started as a very esoteric, very fundamental question, like can you make parthenogenetic mice, has a huge impact broadly. So let me explain what parthenogenesis is. It's development of an egg without fertilization, so they don't have any paternal contribution. So the question was, can we do this in mammals? Can we get development of a mammal, and in our case a mouse, to develop without fertilization right up to adulthood? So I was working on this with my friend Matt Kaufman, and we could develop these embryos, and occasionally we could even get them to develop up to mid-gestation period, halfway through development, but they wouldn't go further. What we did was to make 3 different types of one-cell eggs. In one, we had 2 paternal derived genomes. In one, we had 2 maternally derived genomes, and the third was a control, it's both. We found that the embryos with 2 maternal genomes had a relatively good embryo with very poor placental tissue, whereas the ones with 2 paternal genomes had very poor embryo but relatively better placental tissue. So we got an answer that, there was something that was happening, this non-genetic information that was coming in. And it was not clear how to describe this. And so when I wrote my paper in 1984, I decided to call it genomic imprinting. In other words, the idea was there was this non-genetic information, if you like, which was imprinted on these parental genomes, which was dictating their functions. We started to think that DNA methylation could be the imprint coming from with eggs and sperm. It's clear that this whole process must be occurring during the formation of the lineage which gives rise to sperm and eggs. One of the key changes that occurs at this time is the erasure of imprints. So by the time these germ cells, primordial germ cells, get into developing gonads, imprints are erased. So the slate is wiped clean. It's almost like wiping out a computer disk. You're going to put on a new program, you wipe out the disk. Now, the embryo can develop as a male or a female. And so they have to put appropriate marks depending on whether the embryo is going to become male or female. And you cannot do that without wiping out the disk or the imprints from these germ cells. And by the time they get into the developing gonads, we call this epigenetic ground state, which means that this is the state where suddenly all the DNA methylation is virtually wiped out. The question for me now, which I've been struggling with, is on the evolution of genomic imprinting. I've been wondering whether genomic imprinting has an impact on allowing this diversity of form that you see in mammals. So that's the question that I'm interested in.